Imagine a world where parents could choose their baby's eye color, intelligence, height, or even immune system strength. Imagine a future where genetic diseases no longer exist because we can remove harmful genes before a child is born. It sounds like science fiction, but this is not just a fantasy anymore. Scientists have developed a revolutionary gene editing tool called CRISPR, and it is changing the way we think about human genetics, medicine, and even the future of our species. So, what is CRISPR, and how does it work? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Essentially, it is a natural defense system found in bacteria that scientists adapted to edit genes in any living organism. Think of CRISPR as molecular scissors that can cut DNA at very precise locations. Once a gene is cut, scientists can either remove it, replace it, or even insert a new sequence. This allows for an unprecedented level of control over the blueprint of life. Unlike older genetic engineering techniques, CRISPR is fast, efficient, and relatively easy to use. It has already been applied in agriculture to create crops that resist pests, diseases, and drought. But its most controversial potential lies in humans, especially embryos. Why is this such a big deal? Because CRISPR allows us to edit human DNA at its earliest stage, even before a baby is born. In theory, we can prevent genetic diseases like cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and even some forms of hereditary cancers. Imagine a world where children are born without these lifelong struggles, a world where genetic disorders are eradicated. This potential is extraordinary. It promises a future where suffering caused by genes could be dramatically reduced. But with such power comes enormous ethical concerns. Editing human embryos is not just about helping one individual, it affects all future generations. When we change a gene in an embryo, that changes germline, meaning it will be passed on to that person's children, grandchildren, and beyond. This is a permanent alteration to the human gene pool. Scientists around the world have debated whether we should cross this line. Could CRISPR be used to create so-called designer babies, where parents choose physical traits, intelligence, or even personality? The implications are staggering. It raises questions about fairness, equality, and even the definition of humanity. The first real-life example of CRISPR babies shocked the world. In 2018, a Chinese scientist, He Jiankui, announced that he had created the first genetically edited human babies. His goal was to make them resistant to HIV by altering a gene called CCR5. While some praised the potential scientific breakthrough, the global scientific community condemned the experiment. It was conducted in secret, without proper oversight or ethical approval. Many worried about unintended consequences. Editing genes in embryos can have unpredictable effects, sometimes creating new problems instead of solving them. Beyond ethics, there are technical risks. CRISPR is precise, but not perfect. Scientists call the mistakes off-target effects, where the molecular scissors cut the wrong part of the DNA. This can lead to serious health problems or genetic disorders that didn't exist before. Imagine trying to remove one disease and accidentally causing another. The stakes are unimaginably high because we are dealing with human lives, and the effects are permanent. Despite these concerns, research continues. Scientists are using CRISPR in adults to treat certain diseases, such as sickle cell anemia, some cancers, and eye disorders. These treatments are temporary and affect only the individual, not their future children, which avoids the ethical dilemma of germline editing. But the lure of editing embryos remains strong because of its potential to completely eradicate genetic diseases before a child is even born. The ethical debate isn't limited to health. Imagine a future where parents could select traits like height, athletic ability, or intelligence. What would that mean for society? Could it create a genetic, elite, class, increasing inequality and social tension? Could humans become obsessed with perfection, treating their children like products to be designed instead of lives to be nurtured? Bioethicists warn that we must tread carefully. Once germline editing becomes commonplace, it may be impossible to reverse. There are also cultural and religious considerations. Many societies believe that human life is sacred and should not be altered at the most fundamental level. Others argue that preventing suffering is a moral obligation, making CRISPR a tool for good. Balancing these perspectives is complex, and international regulations vary widely. Some countries have banned germline editing, while others are still deciding. Despite the controversy, CRISPR represents a monumental leap in science. It has already transformed genetics, agriculture, and medicine. It forces us to ask uncomfortable questions about the limits of human intervention in nature. It challenges our understanding of ethics, responsibility, and what it means to be human. The technology itself is neutral. It is our choices that determine whether it will benefit humanity or lead to unforeseen consequences. So, can we edit babies using CRISPR? The answer is yes. The technology exists, and we have already seen real-world attempts. But the bigger question isn't whether we can, it's whether we should. Do we have the wisdom, ethics, and foresight to use this power responsibly? Are we ready to face the consequences, not just for one child, but for generations to come? In the end, CRISPR is not just about science, it's about humanity's future. It is a tool that could eliminate suffering, but it could also redefine life as we know it. Every decision we make today about this technology will echo far into the future. The debate is ongoing, the risks are real, and the possibilities are both thrilling and terrifying. As we look ahead, one thing is clear: CRISPR has opened a door we cannot close, and the world will never be the same again.